Cool. It is recording now. Awesome. All right. Hi, everyone. Welcome to the Quantitative Seminar series. It's going to be virtual for this fall quarter, so thanks, everyone, for tuning in to it. So I'm Brielle, and I'm one of the organizers for this year's Quantitative Seminar series for the Converse Lab, and I will begin with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes in the Suquamish, Tulalip, and Muckleshoot nations. Our lab also acknowledges that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. So Abby asked for a short little introduction and will be introduced as such. Abby is a master's student in the University of Washington's Quantitative Ecology and Resource Management program. She is a Seattleite through and through, having been grown up here, earning a BS in statistics from the University of Washington in 2018. And the work she will describe today is degradation in mark-resight studies, problems and a proposed solution. And this is a portion of her master's bypass. And Abby would like to take questions after the talk, so we can use the questions for after the talk. And thanks everyone for coming out today. And thanks for our speaker, Abby, and for sharing your work with us. And with all that, it's my pleasure to turn it over to Abby.
Awesome, thanks Brielle. Let me remind myself how to get the screen sharing set up and then we'll get started.
Cool.
Can you guys see that?
Yes, I can see it, we're good.
Excellent, thanks. All right, so good afternoon, everybody. I hope everyone had a wonderful summer and I'm thrilled to be the first presenter kicking off this year's quantitative seminar. So my name is Abby Bratt, and today I'll be discussing mark degradation in mark-resight studies, both the problems that it poses for estimating demographic rates like survival, and one proposed solution. So I'll get right into things with a quick introduction to capture mark-resight models, and then I'll talk a bit about what happens when the assumptions of those models are violated, and in particular, the focus of this talk, partial mark loss or degradation. I'll then describe our proposed model and the algorithm that's central to this approach using a worked example and a custom MCMC sampler. And finally, I'll present some results from a simulation study using the described model and a case study of a local threatened population. And that sounds like a lot, but I'll leave some time at the end for discussion and I'll be happy to take any questions you may have then. So the types of models that I'll be talking about today are called capture mark-resight models. And I first want to make sure that I differentiate between capture mark-resight and capture mark-recapture. So as the name implies, in both study designs, you start out by placing a uniquely identifying mark on a subset of your population of interest. So for instance, consider the bird in hand here, which is a streaked-horned lark chick. And it's just received 4 bands in total, a unique combination of 3 colored bands and 1 metal USGS band that has a unique number on it. So if we were doing a mark recapture study, we might only put the metal USGS band on these guys. And in that case, we would have to physically recapture the individual in order to identify it. In contrast, in mark-resight studies, at least some of the marks you deploy are field readable, like the colored bands here. And so then you don't need to physically recapture the individual to identify it. There are a lot of advantages to this type of study. They're less invasive, more cost-effective, and these data and the models that we use to analyze them are the focus of my talk today. So capture mark-resight models are a class of state space model. So that means they have a latent state process and some observed measurement of that process. So for example, let's say we have an individual animal that is captured and marked on occasion 1. We know it was alive at this point because we had it in the hand. So there's a deterministic relationship between the true state and the observed state at this point. After that, the individual survives according to some stochastic process, and we can either detect it or not according to another stochastic process. Once the individual is dead, it stays dead, and we necessarily do not detect it. So the diagram shown here represents a simple form of this model that has just two states. You're either dead or alive. And it's primarily used to estimate survival. But we can imagine more complicated multi-state versions of this model that could estimate things like movement between sites, infection rates, and things of that nature. So for instance, I'll be talking a lot today about multi-site models, where individuals can move around a discrete set of sites and subsequently be detected at those sites. So take the same individual trajectory and observation process, but now our observations are indexed by their location, where the individual was observed at site 1 on occasion 1 and site 2 on occasions 2 and 3, or rather just occasion 3. Now, there are quite a few assumptions in these types of models. The ones most relevant to what I'll be talking about today are the so-called marking assumptions. So those are that animals are correctly classified as marked or unmarked, that individual marks are correctly and completely identified, and that no mark loss occurs. But there's quite a few ways that these assumptions can be violated, and a lot of really smart people have spent time thinking about what to do in these cases, of which I'll introduce two now. So I'll be talking a little bit about mark misidentification and complete mark loss. So first consider mark misidentification, and that's when observers attribute a mark to the wrong individual. So in this case, what's called the encounter history, which is a record of which individuals were recorded at each time step, would contain an error. So let's say I had two individuals, one with a blue-green mark and one with a blue-purple mark. Now those marks are really similar, so they might be easily confused in the field. So now let's say that the blue-green individual was detected at occasion 1, which is indicated by a 1 in the encounter history matrix, and not detected on any other occasion, indicated by the zeros in the matrix. But let's say observers misidentified the blue-purple individual as the blue-green individual, resulting in the encounter history shown here. So now in the context of the model, this encounter history implies something different about survival and detection probabilities than the true encounter history does, and therefore it introduces bias into model estimates. In the case of mark misidentification, Tucker et al. Proposed one approach, which was data filtering to reduce the error rate within a data set. Or Roche et al. Proposed the double marking approach to estimate the frequency of mark misidentification and therefore adjust for associated bias. Now let's talk a little bit about complete mark loss, which is another possible problem in mark-resight studies. Complete mark loss essentially removes an individual from the data set because it effectively can never be encountered again. So consider the same two individuals as before with the same true encounter history. But now, say the blue-purple marked individual loses that mark after the first occasion. Then when an observer re-encounters it on subsequent occasions, it looks like an unmarked individual and does not get recorded. So this results in both bias and loss of precision in survival estimates, as demonstrated by Arneson and Mills. And one common way of addressing this issue is again with double marking, to estimate the frequency of mark loss and adjust for the associated bias. However, one area that has not received much attention in the literature is partial mark loss, which is sort of a hybrid of mark misidentification and complete mark loss, and is the focus of my talk today. So now let's say we've got the same two individuals, same true encounter history matrix, but now the blue-purple marked individual drops its purple mark after the first occasion. Now in this case, it's still detected on subsequent occasions, but it's recorded as a blue mark. But we know we didn't deploy a blue mark as part of our study, so every observation of this blue mark does not map nicely to a unique individual. It could be the blue-green marked individual on some occasions or the blue-purple marked individual on others, depending on the latent processes governing who's alive and who was truly detected. So the reason I started thinking about this problem is because of my study species for my master's work, the streaked-horned lark, pictured here as a chick. And it's freshly banded, as a lot of songbirds are, with a unique combination of 3 colored bands that are field readable and a metal USGS band that's not field readable in most cases. So when these birds grow up, ideally they'd look like this. So notice that this bird I'm circling it with my cursor if you can see that, has retained all 3 colored bands and its metal band. So if you saw this bird in the field, you would be able to completely and correctly identify it. But these birds are very pesty, and quite often you'll see something like this. And apologies for the photo quality, I took this one through a spotting scope. But if you look closely right here, you can see that the bird only has 1 orange band left in addition to its metal band. So this bird could be any bird that had received an orange band as a chick, which leaves a lot of options. Now at this point, this may seem like a hyper-specific problem, but as I started reaching out to other researchers, it's actually quite widespread across both taxa and mark types. So for instance, here's an example of a red knot, which is a shorebird, with alphanumeric tag AX3 that's clearly field readable, and here it is in 2012. Now here's the same bird in 2020, and the flag is visible, but it's no longer field readable. So to individually identify this bird, they had to physically recapture it. Here's another example, this time of a sanderling. Here it is in 2016, and it's got a ton of bands on it to uniquely identify it. It's got a metal band, orange band, a flag, a light blue band, and another orange band. Now here's the same bird in 2018. It's now missing the flag that it had before. And here it is this year, missing both the flag as well as one of its orange bands. And now I know this isn't necessarily a super birdie crowd, so I also brought a couple of examples of this problem in other taxa. So here's a polar bear. It's common in studies of some mammals to tattoo them with a uniquely identifiable code, which you can sort of see here. So we can see that this bear, has been tattooed, but the tattoo has faded beyond the point of recognition. And we could imagine something similar happening in marine mammals like sea lions, who get branded at a young age, and then as they grow, the brand warps in shapes and size and becomes unreadable. Here's a turquoise darter. Rachel Katz et al. In 2015 performed a capture mark recapture experiment on these guys using these fluorescent elastomer tags. And they used unique combinations of tag color and body position to mark individuals. But they ran into this problem when the tags would move around inside the body cavity of the fish, and that would make the mark ID uncertain. And this is my last example, a bicolored striped sweat bee. So Nick Dorian was studying the site fidelity of bees to urban gardens. And you can sort of tell in this picture that the bee on the left has some paint remnants on the thorax, the green bit near the top of the bee. And they were using a combination of paint colors and locations to uniquely identify bees. But they noticed that in some cases, the paint would slough off in such a way that you could tell the bee was marked, but not who it was. Now, I just want to take a moment to quickly acknowledge that this problem has received some recent attention in the literature on closed populations, where capture-recapture, and in particular spatial capture-recapture, is used to estimate population density, like in this paper by Augustine et al. But this problem is pretty different in the context of an open population because there's that additional uncertainty at each sampling occasion about the true state of an individual. So now I'll describe the model that we've developed to deal with this problem. And recall that we're using a state space framework where we have a latent state process, which is denoted Z here. And the state of individual I at time t is conditional on the individual's previous state and is distributed according to a categorical distribution using probabilities drawn from state transition matrix psi, where there are s plus 1 possible states. And this state transition matrix can have a lot of different structures that are specific to the model. But here's one possibility for a multi-site model, where each site represents a unique state, and there's an additional dead state. So this model incorporates between site movement, where individuals have some probability of staying at their home site along the diagonal, these phi11, phi22, phi33s. Or alternatively, individuals can transition to a new state these off-diagonal fees. Right. And we could put whatever model we want on these off-diagonal fees. One that might make sense and one that I use in my talk today is these are probabilities decay with the distance between, for instance, sites 1 and site 2. Individuals survive at rate phi, die with rate 1 minus phi, and once you're dead, you obviously stay dead, so with rate 1. On top of this latent state process, we have the individual observation process Y, which is also latent in this case. It's conditional on state Z and also categorically distributed using probabilities drawn from transition matrix pi. And once again, there are many possibilities of structures that you could place on the observation process, but the simplest and the one we use here is just if you're at site S, we detect you with probability P. If you're at site R, or if you're at site S, we detect you at all other sites with probability 0, or you're not detected with probability 1 minus P. Now, thus far I've described a pretty standard multi-state model, but here's where things get a little bit interesting. So we introduce an additional latent process, which is the mark degradation process, and that's represented by M here. So that is an individual's mark at time t is conditional on the mark it had at the previous time step, and it's categorically distributed using probabilities drawn from some mark transition matrix, theta. The mark transition matrix is specific to the set and type of marks deployed, but if you know that, then you can generate theta programmatically. So for example, let's say we are marking birds using colored bands like I showed before, and we have 3 birds in our population. One of them gets a blue-green mark, one gets a red-green mark, and one a blue-purple mark. Now let's assume that they drop those individual colored bands with rate beta, and we're also going to assume that they are identifiable as marked, even if they drop all their colored bands. We denote this case N, and it's analogous to the case where a bird would drop all its colored bands but retain its metal GS band, which is really, really uncommon for them to lose. So in this case, the mark transition matrix would be as follows. The set of all possible observable marks is blue-green, red-green, and blue-purple. Those are the marks we deployed. But we could also observe just green, just purple, just blue, just red, or no colored bands at all. Now, working through the first row of this matrix as an example, if an individual starts in mark blue-green at time t, it could stay in that mark at time t plus 1 with probability 1 minus beta squared, drop either its blue band or its green band with probability beta times 1 minus beta, or it could drop both colored bands with probability beta squared. Okay. So I want to emphasize at this point that everything I've described so far that's indexed by individual, the state, late observation, and mark degradation processes are all latent. And that's because we don't actually observe individuals. We observe marks, which may or may not uniquely identify individuals. So we need to somehow connect these latent states to the data that we actually observe. And we do that by introducing this array called Y-multi. It contains ones if individual I is in mark K at site S at time t, and zeros otherwise. And this array is deterministically related to our data, Y-obs, by marginalizing over individuals. So that is, the data we observe are counts of mark K at site S at time t. And I know that's a lot, so let's unpack it a little bit more by looking at it visually. Just forget about space for now. So now Y-multi is a 3-dimensional array representing whether or not individual I was in mark K at time t. If we marginalize this array over the mark dimension, we obtain the data that we want, which are encounter histories of individuals across time. If we marginalize this array over the individual dimension, we get the data that we have, which are counts of marks observed across time. So this latent data array is how we connect the latent individual observation process to the observed data. And now we've got a model. So it seems like we're good to go ahead and run it. In fact, we are close, but actually there's one more challenge ahead. So we want to fit this model in a Markov chain Monte Carlo or MCMC framework. And if you're not familiar with MCMC, this is a very, very short summary of how it works. I like to think about it as a complicated system of guess and check, wherein you have some unknown parameter or parameters that you'd like to estimate. So the first thing you do is you propose a possible value for those parameters. Then you evaluate the likelihood of your model conditional on that parameter. Then you propose an alternative value for those parameters, evaluate the likelihood again, compare those likelihoods, and based on that comparison, you choose one of the proposed parameter values to save. That saved value then contributes to your overall estimate of the posterior distribution for the parameter, which is what the GIF at right is showing. So what's called the trace plot is on the bottom. That shows how your saved parameter is jumping around the parameter space and getting contributed to the posterior distribution, which is shown on the top. So if you repeat this for a long time, you can get pretty good estimates of your parameters. And generally, the hard part of MCMC is figuring out how to cleverly propose alternative parameter values. And it turns out to be particularly difficult in this case. So recall that the latent observation process YIT is categorically distributed. And therefore, we might, or a computer program might, by default, put independent categorical samplers on each node of YIT. So for instance, if we've got 3 individuals monitored over 4 occasions, then there will be 3 times 4 equals 12 unique categorical samplers on each element of Y. But sampling Y in this way ignores the structured relationship that we just explored between the observed marks and latent encounter histories. And now we could run the model this way using those independent categorical samplers. And the magic of MCMC is that if we ran it infinitely long, we would be guaranteed that the model would converge. But obviously, we don't have infinitely long, and leaving the default categorical samplers on these Ys is going to be painfully, unimaginably slow. Because the probability that you, by chance, propose YITs that are valid, conditional on all other YITs, is very, very low. So for instance, if we proposed that the red-green individual was detected at site 1 on occasion 2, but no matching marks were observed at that site or occasion, the model is going to reject that proposal. So in short, we want to minimize the amount of time we spend making proposals that we know the model is going to reject because they're just infeasible. And so the point being is that if we somehow acknowledge the structured relationship between observed marks and latent encounter histories, we can sample Y much more efficiently. One way to do this is by using what's called a block update. So instead of updating each element of Y one at a time, we can perform these big clunky updates of the entire Y matrix at once. The problem with this is that the computer doesn't know how to generate encounter histories that are valid, conditional on the set of marks we observe. So we need to generate those valid encounter histories manually. And the way that we chose to do that here is by using a backtracking algorithm. So backtracking algorithms are a type of recursive tree search algorithm that are commonly used to solve constraint satisfaction problems. So for instance, if you were trying to tell a computer how to solve a Sudoku puzzle, you might use a backtracking algorithm to iteratively build the solution by testing out numbers 1 cell at a time. So backtracking algorithms build unique candidate solutions incrementally, and they abandon candidates, or backtrack, hence the name, as soon as it's apparent that a candidate solution will not lead to a valid solution. It's a pretty fast algorithm relative to other exhaustive search algorithms. And in fact, it's guaranteed to find all solutions within a bounded amount of time. So I'll quickly work through some pseudocode to give you a sense of how this algorithm works, and then we'll step through a worked example using our model. So here, let P be the data representing the problem at hand. In our case, it's the set of observed marks on any given occasion. And then let C be a node of the search tree representing a partial solution to the problem, i.e. a partial mapping of observed marks to individuals. Then we can define some procedure we'll call reject that returns true if and only if the partial solution C is not worth completing. So in this case, that would mean that individuals have been assigned to observed marks such that at least one mark has no possible individuals to which it can be assigned. If this happens, the algorithm backtracks, meaning it stops exploring this solution's branch of the search tree. We also have some procedure called accept that returns true if C is a complete solution. That is, that all observed marks are mapped to individuals. And if this is true, we want to save solution C. If C is neither accepted nor rejected, that implies that it's incomplete. And therefore, we use this procedure called first to make an extension of the candidate C. And we're going to call that partial solution S. So in this case, conditional on the set of marks already assigned to individuals at node C, we're going to assign another individual to a mark. Now, here is the recursive bit. So now S forms the root node of a new subtree, which we'll then explore using the backtracking algorithm to continuously generate and test extensions to S. And I know that was really abstract, so let's take a look at a worked example in the context of the model I've described. So let's return to our trusty example population of 3 marked birds. We've got blue-green, red-green, and blue-purple. And now let's say that they're monitored over 4 occasions at 3 different sites. So we've got a multi-state model where state 4 represents dead in the state transition matrix and not observed in the latent observation matrix. 1 through 3 represent sites 1 through 3 respectively. And then matrix M represents the mark state. So focusing on the combination of encounter history and mark state, let's take a closer look at what exactly is going on here. In year 1, we banned the individuals, so we know exactly who everyone is and where they are. BG's at site 1, red-green's at site 2, and blue-purple's at site 3. Then in year 2's resites, we observed 2 blue marks, one at site 1 and one at site 3, in addition to the red-green mark at site 1. In year 3, we've got a blue mark at each of site 1 and site 3 again, and a green mark at site 2. And then in year 4, no one was observed. So notice here that on equations 2 and 3, when we observe those blue marks, we don't know for certain which is individual blue-green and which is individual blue-purple. It's probably more likely that individual blue-green stayed at site 1 throughout and blue-purple stayed at site 3 rather than the 2 birds switching back and forth. But again, there's some uncertainty there. Okay, so to demonstrate the backtracking algorithm, let's take the set of marks observed in year 3. So we've got that blue at site 1, green at site 2, blue at site 3. And now we'll build our first candidate solution, which we do by assigning the blue mark at site 1 to the blue-green individual. Now this solution is obviously incomplete, so we then increment it by assigning the red-green individual to the green mark at site 2. And finally, we complete the solution by assigning the remaining blue mark to individual blue-purple. And there we go, that's one valid solution. So now returning to the root node, which is just the mark not assigned to any individuals, well, let's explore the remainder of the tree. So this time, let's assign the blue at site 1 to individual blue-purple. And incrementing, we'll assign the green mark at site 2 to individual blue-green. But there's a problem here. Now we only have one remaining mark, which is a blue, and just one unassigned individual, which is red-green, who obviously does not have a blue mark. And therefore, this partial solution is invalid. Now the algorithm is going to backtrack to the last valid partial solution and explore alternatives. So instead, let's try assigning the green mark to the red-green individual and incrementing one final time, assigning the other blue mark to the blue-green individual. And now we've found the only two valid matchings of observed marks to individuals at this occasion. Now, if we ran the backtracking algorithm as described on every occasion, we would get the valid matchings of observed marks to individuals at each occasion. And if we let each of these solutions be a node and build a graph between them that connects each solution from occasion 1 to each solution from occasion 2, and so on and so forth, as is demonstrated here, then each pathway through this graph results in a unique matrix Y. So for instance, the path indicated by the darker arrows here results in the true encounter histories wherein individual blue-green remains at site 1 and blue-purple remains at site 3. So now that we've found all the possible latent encounter histories, what are we going to do with them? And this is where a custom MCMC sampler comes in. So just as a reminder, I noted before that the hard part of MCMC sampling is smartly proposing parameter values. You want to fully explore the parameter space, but you also want to propose values that are feasible. And the way we do this is as follows. So we take the initial log probability of the current matrix, Y. Then we subset the list of all possible encounter histories as generated by the backtracking algorithm, which we've fed to the model as data. And we subset them to only those which are valid conditional on the current state of the model. So in our case, this means that the encounter history can't indicate an individual is detected when the model thinks it's dead, and it can't indicate an individual is detected at a site other than the site it's really at. So basically these Ys have to be consistent with the Zs. So then to propose a new Y matrix, we pick one of those valid Y matrices at random, and then we compute the log probability under this new proposed matrix. Now taking the difference of those log probabilities gives us the log Metropolis-Hastings ratio, which we then use to either accept or reject the proposed encounter history. And whichever encounter history prevails gets saved in our MCMC output and contributes to the posterior distributions. All right, so that's the end of all the abstract stuff, so thanks for bearing with me. And now let's get into some results, see how this thing works. So we performed a simulation study in which we simulated 50 individuals monitored at 3 sites over 5 occasions. And for each combination of the following parameters, we simulated 100 capture mark-resight data sets. So for survival, we simulated both a high survival and low survival case, and likewise for detection. And we varied beta, that rate of partial mark loss, from 0, which would be perfect data, to 10%. And we also compared 2 mark deployment schemes, one sequential and one random. So what do I mean by that? Sequential deployment is where individuals banded closely in time and space get very similar bands. So for instance, if I was banding chicks in the nest, I might give 3 siblings blue-blue, blue-green, and blue-purple, respectively. And this type of thing is quite common in mark-resight studies, but you might expect it to pose problems because if any of them were to drop that second band, they could all be confused. They would all just have mark blue. In contrast, in random mark deployment, I might give the same siblings red, orange, yellow, green, sorry, that yellow is really hard to see, and blue, purple. Now in this example, any of these birds could drop either band and still be uniquely identifiable. So obviously this is a non-random example, but I'm sure you get the point. Going back to the simulation study, we did all this assuming that individuals had high site fidelity and using a typical passer and banding scheme, which includes unique combinations of 3 colored bands and a metal USGS band. So given all these simulated data sets, we ran 2 models. First was the model described here, which incorporates those observations of partially marked animals. And second was a quote unquote standard mark-resight model, capture mark-resight model, where observations of partial marks are omitted from the analysis. And this omission of partial mark observations is one of two ways that data like this are typically treated, and the other being a kind of a post hoc assignment of individuals to marks. So since we were primarily interested in obtaining estimates of survival, those are the results I'll show here. So we measured model performance according to both the bias and survival estimates, as well as the root mean squared error of the estimates. I'm only showing the survival estimates here because there wasn't a super strong pattern in the root mean squared error. But basically, if a model is reliably producing unbiased estimates, we would expect the bias to bounce around 0 and the root mean squared error to be near 0. So what I'm showing here are the results of the simulation study when partial mark observations are omitted. The left panel has the results in the case of sequential mark deployment, and the right panel random mark deployment. But they're pretty much the same, so let's just focus on the left panel. And the plot shows the relative bias of the survival estimates with respect to true survival on the bottom x-axis, detection probability on the y-axis, and the rate of partial mark loss along the top. So notice that we consistently underestimate survival, with the effect growing stronger as the rate of partial mark loss increases. And this effect is also worsened when both survival and detection are low. Now, this makes some intuitive sense because when we omit observations of partial marks, individuals who undergo mark loss can effectively never be observed again. And the model basically thinks of them as dead. I'll just describe what we saw for root mean squared error. It was pretty much invariant with respect to the rate of mark loss, but it was high when both detection and survival are low, and low when detection and survival are high. And as I said, that pattern remained consistent throughout all the results that I'll share today, so I'm just going to present the relative bias in survival. So keeping these results in mind, let's look at the results for the case when partial mark observations are included. Now, ignore the top panel for now. I'll explain that one in a moment. But looking at the bottom panels, they're laid out just as before, with sequential deployment on the left and random deployment on the right. And the plot shows relative bias of survival estimates again, with respect to true survival on the bottom x-axis, detection probability on the y, and the rate of partial mark loss along the top. So notice now that this model produces estimates of survival that are unbiased on average, which is exactly what we hope to see. All right, now let's return to the top panel. This panel shows what I've been calling the confusion index, which I define as the average number of individuals to which an observation of a marked individual can be associated. So for example, in a marked population with no mark loss whatsoever, the confusion index would be 1. Every observation uniquely identifies an individual. On the other extreme, if batch marks are used, are used, where every individual in a marked population gets the same mark, the confusion index would be the number of marked individuals, because every observation could be any individual. So therefore, the confusion index is basically a relative measure of the complexity of a partial mark loss data set. Note here that these results are shown on a log scale, so the average confusion index becomes quite high as the rate of band loss increases, and especially when the marks are deployed sequentially, as shown on the left. So why does this matter? Why am I telling you this? So it gets at an ongoing challenge with this approach, which is limited computing power. Basically, as the confusion index increases, the number of possible encounter histories can explode really, really quickly. And when this happens, the backtracking algorithm is less and less likely to run because it requires quite a bit of memory. So basically, as the rate of mark loss grows, the percentage of time our proposed model actually runs goes down. And obviously, that's not great. I'll discuss this a bit more at the end, but if folks have any clever ideas about how to deal with this, I would be super keen to hear them. Moving on, we also ran this model using a capture mark-resight data set from an endangered local population of Oregon Vesper sparrow, which is pictured here. And there's a small population of these guys in the South Puget Sound region, monitored on 5 sites, 4 of which are native prairies on Joint Base Lewis McChord, which is the blue bit on the map, as well as Sanderson Airport, which is just outside of Shelton in the northwest there. And this is a difficult species to monitor because there are so few of them, and they're also really sneaky about revealing their nests. But there's still been a lot of intensive monitoring of these guys and a focus on prairie restoration in South Puget Sound. Still, though, relatively little is known about their between-year survivorship and the site fidelity of Oregon Vesper sparrow. So this data set includes 134 individuals marked and monitored between 2016 and 2020, where each individual is banded with between 0 and 3 colored bands in one of 10 unique colors, as well as a metal USGS band. And for the most part, birds were banded in the nest according to a sequential marking scheme. So siblings would get very similar band combinations, as I described before. And in the same period, there have been 62 resites, which include 2 sets of 2 individuals that received the same band, and at least 3 individuals observed with degraded marks. So that means that about 10% of the resites are problematic, which is tough in a small population where every piece of data really counts. So let's get into the results. Here are the estimates of age-specific survival from both models, with partial observations omitted on the left and included on the right. And now you'll notice that the estimates are ever so slightly higher when those observations are included, which we would expect given the results of our simulation study. Juvenile survival is estimated at about 37.5%, and adult survival roughly double that at about 75%. And this is pretty high relative to other songbirds, which is at odds with results from the breeding bird survey, which would indicate pretty drastic range-wide decline of the species. But results like this help us isolate which life history stage is limiting population recovery so that we can better target conservation efforts. So in this case, some other reasons for this decline could indicate either low nesting success or low post-fledgling survival. Now looking a little bit at movement, note that we've got a small sample size, so these results should be taken with a grain of salt. And I know we've got 8 panels here, so let's break it down a little bit. Results from the model omitting observations of partially degraded marks are shown in the 4 panels on the left, and results from the model including those observations are in the 4 panels on the right. The top panels show the age-specific probabilities of staying at your home site, with adults on the left and juveniles on the right. Oops, no, excuse me. Juveniles are on the left and adults are on the right, sorry. Now the bottom panels show the site-specific dispersal probabilities. So what's the probability that you move to a new site? And what you want to look for in this figure overall are differences in color between each of the left 4 panels and the right 4 panels. And what you'll notice in general is that the estimates of movement are higher in the model that includes partial observations. And that's because this data set includes more instances of dispersal movements, particularly for juveniles. So for instance, there's a very low probability of hatchier birds dispersing at these 3 sites. There's a slightly higher probability of them dispersing when those partial observations are included. Similarly, as you would expect the movement probabilities to go up, the fidelity probabilities go down. So whereas there's a very high likelihood of staying at your home site for juveniles when those partial observations are omitted, goes quite a bit down when the partial observations are included. So I'll wrap things up by describing a couple takeaways, lingering challenges, and next steps with this work. As I described at the beginning, complete mark loss and mark misidentification are still likely sources of additional bias. So perhaps some hybrid approach combining this model, data filtering as suggested in the Tucker et al. Paper, and double marking is useful for species that exhibit high rates of mark loss and/or are difficult to observe. Another thing to consider is that loss of marks are likely not independent. As some recent work by Schwartz et al. Have shown, Some individuals are just more likely to lose bands than others, and those individuals are more likely to lose multiple marks at a time. But there are ways to deal with this within the context of the model described. Both the model and the sampler are highly customizable to alternative data structures like Poisson observation processes, the inclusion of individual covariates, et cetera, et cetera. And finally, what I've described today has three main components, each with their own challenges. So the first was the development of the hierarchical model incorporating observations of partial marks. The second was the use of the backtracking algorithm to generate all possible encounter histories conditional on those observed marks. And the third was the development of a custom MC sampler to run said model using said encounter histories. So both the model and the sampler work pretty well. But the backtracking algorithm, on the other hand, remains a challenge. And as I said before, the biggest drawback with this approach is computing power. Because even with relatively small data sets and low rates of band loss, there can be thousands and thousands of possible encounter histories. And running the backtracking algorithm can quickly exhaust even half a terabyte of RAM. Now, I'm a quantitative ecologist, not a computer scientist, so I am quite sure that someone other than me could figure out a more efficient way to generate all those possible encounter histories given the set of marks observed. And I think that this is an important area for future research and for cross-discipline collaboration. So I'll leave you all with a couple of recommendations for how this work can be applied in practice. Now, first, I think you all know that there has to be some amount of synergy between sampling design and modeling approaches. No complex statistical model can overcome data which violate model assumptions. So we recommend doing some kind of stress testing of marks prior to deployment. How do they hold up to weathering or a really pecky bird beak? We also recommend deploying marks in random sequence. In the event of partial mark loss, there's going to be less confusion about who is who if similar marks are deployed randomly as opposed to sequentially. And failing stopping mark loss at the source, the model I've described here works reasonably well for small data sets, but runs out of memory on large ones. One option, though, if you've got a large data set in which partial mark loss is a problem, would be to omit observations of partial marks from the analysis. Now, in this case, you know you would underestimate the true rate of survival or dispersal or whatever parameter of interest, but you may be able to estimate an inflation factor for that parameter, say using double marking or an integrated analysis. So with that, I'd like to acknowledge my funding sources for this work, the Quantitative Ecology and Resource Management Program, as well as the Washington Cooperative Fish and Wildlife Research Unit, and my co-authors for this work, Nathan Hostetter and Sarah Converse. I'd also like to thank the Eco Studies Institute for providing the Oregon Vesper Sparrow data. And I'd also like to thank all the folks that contributed their personal photos of animals with degraded marks to this talk. Folks that report animal sightings like this make this work possible. So y'all are awesome. And with that, I'll say thank you so much for having me, and I will take any questions.
Awesome job, Abby. That was so great. I know this problem is super difficult and such a huge feat, and you found a solution and you explained your solution so clearly and gracefully. So thank you for that. Yeah, so in the webinar format, you can ask questions, post them, and I will read them out loud. And also you can use the hand raise feature and I can unmute you so we can hear your voice to have a little bit of kind of feedback, I guess, in person. But yeah, we'll wait as these questions come rolling in.
Andre told me he had to leave early, so it might be a quiet Q&A period.
It's totally fine. Oh, wait, we have a hand raised. Let's unmute Beth. All right, Beth, you should be unmuted.
Okay, can you hear me?
I can hear you.
Excellent. So great talk. I'm curious because I couldn't see some of the simulation results, but is there a point where, I don't know, like in your sparrow example, 10% didn't seem to matter? So like when do you need to really think about mark loss? What's the percent loss that starts to affect parameter estimates?
Yeah, here I can go back to that slide and maybe we can look at it again. It depends a little bit on what your rate of your true detection and your true survival probabilities are. But about 6% is where things really start to go haywire in my mind. Yeah, if your true detection probability is relatively high, then you don't have to worry about it quite as much. Yeah, does that answer your question?
Yeah, so what's high detection probability? Because those all look, oh wait, that's probability of band loss. So what is a high detection probability?
In their simulation study, we used 70%.
And what would low be?
We simulated 30%.
What was the detection probability for the sparrow?
Yeah, it's pretty high because they do pretty intensive nest monitoring. I want to say it's about 50 to 60% for adults and maybe 40 for juveniles. And the estimated true rate of band loss was on the order of 10%. So it's a population that definitely benefits from this kind of a model.
Awesome, thank you.
Thanks for the question, Beth. Mark has a question, Mark Sherrill. I'm gonna unmute you, Mark, if you wanna talk, or I can do it to your question. Let's unmute. Wait. Or maybe I'm not doing it correctly. Well, I will just read it out loud because I am not doing this correctly. Oh, do you want to talk? Oh, great.
We're all live now. I have control. Abby, that was really cool. I may have missed all of this as you were explaining it, and that's all on me because I thought your explanation was actually rather good. But can you help me understand how it seems to me that there's some joint priors here that are not independent of one another, right? You have the banding matrix, so you know for any given site and time what the possible options are for any bird banded there. And based on your movement probabilities, it seems like there's some space that just isn't possible. And I think you got to that with some of yours, but it seemed like that was more from the data side and less from the prior side. And so, like I said, I may have just misunderstood all this, but maybe you can just remind me how that works.
So if I'm understanding your question, are you sort of getting out like how much is space helping, or how much is space helping you?
Space and time, right? Because on any given banding location and time, you have your matrix. So you know, right, there can only be so many options for a given site and time.
Right. Yeah, so like I said, so if you have the bird in the hand, which you do when you capture it and mark it, you know exactly who it is. So on the first occasion anybody is, when everybody's captured, we know who you are and where you are. And so that is, we know what your home site is. Then on subsequent occasions, this model works pretty well when individuals have high site fidelity. And that's because site is pretty informative of individual identity. Like in the example that I showed, it's much more likely that the blue-green individual is going to persist at site 1 and the blue-purple individual is going to persist at site 3. So not every encounter history that we generate is equally likely, but that likelihood is conditional on the true state of the model. Does that help a little bit?
Yes, I think so. Yeah, I think I should probably just ask you this thing. I'm having trouble a little bit with the idea of right, your possible states governed by, you have some known knowns and some known unknowns. And yeah, if there was zero movement, then your banding matrix would give you, for a location, really strong information, right, about what you would expect to see at a location.
Right.
And yeah, so, okay.
But
All right.
again, it depends pretty strongly on the specific set of marks you deploy, because if at a site, this is common practice in banding birds, you would give everybody at site A in year 1 at least one band in common.
Right.
So depending on which bands they drop, you could have zero movement and you could observe a band that is not uniquely identifying a bird and still have a lot of confusion about who's who.
Yep.
So it's conditional on the specific set of bands deployed, which is what makes this so complicated. And it's like kind of a combinatorics problem in addition to everything else that it is.
Yes, and so, and then I'll just follow up really quickly. Have you thought about the more complicated problem, now that you've solved this one rather elegantly, where some bands fade, right? Red starts to look like orange after a while, yellow will start to look like white, or pink will look like white, something like that. Have you thought about that issue?
Yeah, yes, this plagues me. Well, general practice is like there are some band colors that you would want to avoid using, and particularly some band colors that you would want to avoid using next to each other, like a red and an orange or a red and a pink, because yeah, as that band ages, it's going to look really, really similar. In some cases, that's not an option because you're simply trying to band more individuals than you have unique combinations. So in this, like for most pasturins where you've got 10 colors, 3 colored bands, you have something like 1,500 unique combinations. If you band more than 1,500 birds, you're going to have to use some of those crappy combinations. Yeah, so you could put like, you could have a kind of a multi-event model framework where your observation of a band is not, you assume that it's imperfect. Right now we've been assuming that it's perfect, but you could complicate the observation process a little bit.
Okay, cool. This would have helped me 25 years ago. Thank you.
Thanks, Mark. So we have a question from Dan. Let's see if I can do this correctly. All right, Dan, you might be unmuted.
Seems possible.
Awesome.
Great talk, Abby. And ninja level Zeringan skills, right? I never know how you pronounce that, but great stuff.
Thanks.
Yeah, so it seems like, right, there might be kind of a trade-off, right, between like a conventional MCMC is going to take freaking forever jumping around to a bunch of places that can't exist, but every individual draw is very computationally non-intensive. Yep. Whereas with your approach, right, you get rid of all those inefficient draws, but it becomes really computationally intensive. And I'm wondering if you like sketched out the frontier on that, right? Is there some point where it's like, on the one hand, a conventional MCMC might take 2 weeks, but it would run. Whereas like the backtracing approach would just blow up your computer, right? So, runtime is never,
Yeah, that's a great question. Again, it's the trick is it's hard to sketch a frontier like that because it really depends, again, on the bands you deploy. So for instance, and how they're lost, which is a stochastic process. Because the confusion index, if the confusion index is close to 1, you could, and the population size is small, to your point, you could probably use a default categorical sampler on each element of the Y matrix, and it would probably run in a couple of weeks, maybe. Anything higher than about a 1.1 confusion index is gonna take years and also years off my life. So that is where the backtracking algorithm comes in handy.
Got it. Cool. That's a really clever approach.
Thanks.
Thanks, Dan. Okay, we have a question from Jamie. Let's see. Hopefully that worked. Oh. Jamie, I think you're unmuted, but we can't hear you. All right, I will just raise it to someone else then, if that's okay. Sorry, JB. Okay, Gary had a question. All right. All right.
Can you hear me?
Yes.
I can hear you. How's it going, Gary?
Hi, Abby. Great talk. I don't have any questions about the math, but I do have a question about your recommendations. You mentioned recommending randomizing the bands that you're putting on there. I was wondering if that's related to only the color bands or the also including the aluminum band, which you wouldn't ever lose.
Right. Yeah, my understanding is that there's strict rules about how you deploy those aluminum bands. So yeah, no, it's totally fine for those to go in sequence, especially like in a short-lived bird. If you recapture the bird, you're probably going to be able to identify it by that aluminum band, unless like it's super something has abraded it. But yeah, no, it's just the colored bands. So randomizing both the position and the colors in sequence.
Okay, I mean, I was thinking about the situation where we could put them on the left or the right leg.
Oh yeah,
there's some advantage to putting them all on the same leg for one site, because that's often a strategy is you always put the aluminum on the left leg for one site and the right leg for other sites.
Yeah, you can generate more unique combinations if you alter the leg that you put the band on, but it doesn't matter in the same way of like for random deployment. It's just to make more combinations. Does that make sense?
Yeah, I think so. Might pick your brain later about it.
Yeah, absolutely.
Thank you.
All right, thanks. So we have a question. We're going to do two final questions, so we're not going too far over. So we have a question from Simone. I don't know, Abby, if it's easy for you to read the question yourself. I'm going to unmute.
Yep.
Simone, if that's okay. Hopefully I did that correctly. Oh, there we go.
Hey, can you hear me?
Yes.
I hear you.
Yeah. So I wrote it out because I felt like I was going to have a hard time explaining what I meant. But I was just wondering whether your model accounts for backtracking your inferences in a way. So if you gain information in 1 year that you can then use to inform your inferences. Sorry, my husband's also in a meeting in the back. That can then inform your inferences from your last year. So I gave an example where basically in year 2, you didn't capture a bird or whatever, and in year 3 you did. So that would give you information about what another bird who had lost a tag is. Does that make sense?
Yeah, no, that makes total sense and is a really good question. I would have included an example like that in the work example, but it's just so complicated already. So basically, I think what you're getting at is, let's say let's just use your example. So you've got blue, 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 green, blue, yellow. In year 2, you've got blue, blue that's detected as just blue. I think is what's going on, but basically,
right.
Yeah. I think so. Let's say then in year 3, you detect blue, green as blue, green. So then you know that blue mark that you observed on year 2 couldn't have been the blue, green individual.
Exactly.
An important thing to note is that we assume that the mark loss process is generative, that like the only errors that we're seeing are process errors where you're actually losing bands, not observation errors where you just detect one of the bands. Yeah, so when we make the graph connecting all the solutions that you generate from the backtracking algorithm, there's an additional step, which is loop over every unique pathway through that graph and make sure that all the encounter histories would be consistent. So you would exclude any solution that has like a red green assigned to a green mark on occasion 2 and then assigned to a red mark on occasion 3, because that's not possible.
I follow. So I guess in general, my question was just how do you account for that? So I think that's really interesting. Again, like Mark, I wish I had this this method a few years ago when I did my huge capture study as well. So
Cool, thank you. Yeah, that's a great question, and it's kind of an additional step post backtracking algorithm before you can actually run the MC sampler. You want to make sure that all those encounter histories that you generate are valid.
All right, we're going to do one final question, and I'm going to read it out loud. It's from Jamie. Awesome talk, super informative and interesting, and I agree with that, definitely. So her question is similar to Mark's in that I'm wondering if the multi-state nature of the model is necessary for the recursive process to function efficiently enough to have enough computing power to arrive at an estimate, since there is more info available. Or would the model work for a non-multi-state model, such as a simple CGS model?
Yeah, it's a good question. There's nothing stopping this approach from being used on a CGS, like a two-state CGS model, where you've just got states that are dead or alive. The reason that we fit it in this multi-state framework was because we thought that, okay, if you've got a species that's got high site fidelity, you're going to be able to distinguish better which encounter histories are more likely than others, because space is informative of individual identity.
Cool. All right, well, that's all our questions. Thanks everyone for tuning in today. And thanks, Abby, again, for an awesome talk. And you're a brainiac, and it was awesome to hear from you. So hopefully I see a lot of you next week for our second week of the quantitative seminar series. Thanks again. So have a good weekend.
Thanks, everybody.
Nice job.